Welcome to the Scientific Dose. Today we are unpacking the incredibly complex, completely fascinating world of immunology. We're going to bridge the gap from the tiny microanatomy down at the molecular level, straight up to the macro defense system that protects your entire body. We'll be looking at the absolute incredible scope of what our immune cells and proteins actually do to keep us alive, and we're breaking down the molecular evidence directly. It's going to be a wild ride.
Now, at the very foundation of this entire system, we have the interaction between our defender proteins and their targets. So let's get our terms straight. First up, antibody, a protein, specifically an immunoglobulin that binds an antigen. And second, antigen, a substance that is recognized by the immune system. Grasping the nuances of these two definitions? That's the absolute key to everything that follows.
Here is our roadmap for today's journey. Number 1, basic structure of antibodies. Number 2, meet the 5 isotypes. Number 3, antigens and binding sites. Number 4, the power of multivalency. And finally, number 5, lymphocyte antigen recognition.
Okay, let's dive into this with Section 1, basic structure of antibodies. Looking at this fundamental Y-shaped tetramer structure right here, you'll see those red sections at the top. Those are the variable regions, the highly customizable parts that actually lock onto the target. Just below them, those blue sections, those are the constant regions, forming the stable trunk and the lower arms of the Y. And crucially, if you scan the center and the connections between the arms, you'll notice those small black double lines. Those represent the disulfide bonds. They are literally the tough biochemical bridges that physically link the two larger heavy chains in the middle and the smaller light chains on the outside, securing them together into this rigid, highly functional Y shape.
Section 2, meet the 5 isotypes. To keep these 5 unique variations straight, you really just need to remember the gamed roster: IgG, IgA, IgM, IgE, and IgD. Memorizing gamed is honestly the easiest way to recall these specific variations, all of which are secreted by our plasma cells. Comparing these isotypes, you'll see the stats vary wildly. Let's look at molecular weight. We start at 150,000 for IgG, IgD, and some IgA, then step up to 190,000 for IgE, and hit a massive 900,000 for IgM. In terms of normal serum levels, well, IgG absolutely dominates. If you combine its subclasses, it makes up an incredible 65 to 70% of our total serum antibody levels. IgA sits around 3 to 3.5 milligrams per milliliter, IgM at 1.5, and IgD and IgE, they just exist in tiny trace amounts at 0.03 and 0.003 respectively. Next, looking at the in vivo serum half-life, IgG actually lasts the longest, up to 23 days, while IgE only sticks around for about 2.5 days. And then there's a truly phenomenal stat right in the middle, the ability to cross the placenta. Notice how only IgG has the plus signs there? Because it's a small monomer and highly abundant, it physically crosses the placenta to give newborn babies crucial passive immunity before they can even protect themselves. How cool is that?
Now, what's really interesting about this slide is Section 3, antigens and binding sites.
Here we get to see the precise point of contact, the highly variable complementary determining regions, or CDRs. Notice how the blue, green, and red tertiary structures cluster right at the tip of the protein on the lower left. Then, looking at that stunning yellow mesh pocket on the bottom right, exactly 6 of these CDRs, 3 from the heavy chain and 3 from the light chain, come together. They fold and construct these incredibly unique 3D dimensions, creating very specific pockets, grooves, or extended flat surfaces. They act exactly like a perfect custom-made puzzle piece for a very specific antigen. Comparing a resting versus an activated mast cell perfectly shows this binding in action. Inside the resting cell on the left, those dark blue circular granules are completely packed with histamine and inflammatory mediators. The yellow IgE antibodies are anchored right on the outside. Now, since IgE is present in very low serum amounts, as we saw earlier, it's mostly heavily bound here to these outer Fc receptors. Now, look at the right side. When a multivalent antigen, that red strand you see, comes along and physically cross-links 2 or more of those yellow IgE antibodies together, it triggers an immediate chain reaction. The cell activates and literally explosively releases all those blue granules into the surrounding tissue. And boom, that causes an inflammatory or allergic response.
And this brilliantly illustrates Section 4, the power of multivalency. Looking at the structural geometry on the bottom left,
The dimeric IgA links two of those basic Y-shaped monomers end-to-end at their constant regions. Because each Y has two grabbing arms, this dimeric structure yields 4 separate binding sites. But then, looking at the absolute powerhouse above it, IgM. Wow. The body arranges 5 of these monomers into a beautiful, almost snowflake-like pentamer shape. By organizing in this exact geometric ring, a single IgM molecule offers a staggering 10 separate antigen binding sites. This multivalency allows for incredibly tight multi-point binding to a pathogen, effectively sweeping up threats with extremely high efficiency.
Let's move to and see how this builds into Section 5, lymphocyte antigen recognition. On the left side of the maturation pathway, notice those Y-shaped structures embedded directly on the surface of the circular B cells. Those act as direct membrane-bound receptors. Shifting our focus to the T-cell interactions on the right, the pink cytotoxic T-cells and the blue helper T-cells aren't just grabbing free-floating targets. Instead, they are physically locking onto those highly specific, colorful MHC molecules, the little teal and orange clasps that are actively being presented by the central yellow antigen-presenting cell. It's a much more highly regulated handshake, so to speak.
The distinction between these two is pretty stark. B cells engage in a binary interaction, meaning they bind directly to free, soluble antigens, which can be proteins, polysaccharides, or lipids, without any need for an MHC molecule. Furthermore, they recognize accessible, hydrophilic, mobile peptides. T cells, however, demand a ternary interaction. They cannot bind free, soluble antigen at all. They strictly require internal linear protein peptides to be processed and presented on an MHC molecule. The T-cell receptor, the antigen, and the MHC molecule form a strict 3-part complex.
Which brings us to an incredible question to ponder. How do these highly specific variable regions adapt to rapidly mutating viruses? I mean, if our genetic code is fixed, how on earth does the immune system generate millions of completely new, custom-tailored antibody locks for viral keys it has never even seen before? Understanding these microstructures isn't just basic biology. It is the fundamental basis for ongoing human survival against constantly evolving microscopic threats. It literally all comes down to a mind-blowing process called somatic hypermutation, leaving us with a perfect mystery to unpack the next time we dive into the science.